Okay, this will be our Laboratory 23 discussion. So we will go ahead and look at those. So this will be 6.2a and looking at white blood cells. First thing we're going to look at is called the total white blood cell count. In many ways, it's similar to the total red blood cell count. Remember from Lab 22, we looked at the inside squares, 5 of them, counted all of the red cells in each one of those squares, add them up, and multiplied by 10,000. For the white cells, we're going to count all the cells in these outer larger areas that are labeled A, B, C, and D here. We then add those up and multiply by 50. 50 is the dilution factor. These are larger areas, so we need a smaller dilution factor. And so we expect somewhere between 25 and 50 in each one of these. If we had 25 in each, that would be 100 times 50 would be 5,000. If we had 50 in each, then it would give us a total count of 10,000. So I was just looking at that, and one of the things that we need to look at then is, okay, normal white cells, 5,000 to 10,000. What do we need white cells for? Well, remember, white cells are our immune cells, and so that's important. So if you have below the normal amount, given the range is 5,000 to 10,000, that would be lower than 5,000. You would have a disease called leukopenia. Leuko-white, penia means too few, right? So leukopenia is when you have too few white cells. Leukopenia is always bad. It's always pathological. It's never a good thing to have too few white cells. And in these cases, somebody would be, no matter what the actual condition that caused it, this person would be immunocompromised. They'd be much more likely to pick up diseases or have problems with diseases that normally wouldn't be a big deal. So as an example, when we first started seeing people with HIV positive that became AIDS, back in the 80s, we saw them develop diseases that we've never seen before because they became so immunocompromised and their immune system just couldn't take care of these everyday things that we take for granted we can. So that would be leukopenia below 5,000. If you had more than 10,000, that would be called leukocytosis. And leukocytosis could be good or bad depending on what the thing was. If you had an active infection, then leukocytosis would be a good thing because your body would be producing more white cells to help fight that infection, right? But it could be bad based on what the problem was. As an example, you could also have leukocytosis if you had leukemia. Leukemia is a form of cancer, right? And that certainly wouldn't be good. So the leukocytosis depends on what the reason is. Just as an aside, if you had leukemia, and we talked about this in lecture, if you had myeloid production, remember myeloid refers to the bone marrow. If you had lymphoid, it refers to the lymph. All of the white blood cells can be made in the bone marrow through myeloid production, and 2 of the white cells can be made through lymphoid production in lymph tissue, and that would be the lymphocytes and the monocytes. So, because white blood cells help provide immunity, it helps if we understand them a little better. And there's 5 basic types of especially the lymphocytes. There's a whole bunch of subtypes. So we can divide them up into 2 general categories, granulocytes and agranulocytes. The granulocytes are called granulocytes because they have cytoplasmic granules. So they have granules in the cytoplasm. The agranulocytes do not have granules in the cytoplasm. So our granulocytes are the neutrophils, the eosinophils, and the basophils. So notice they all end in that last fill word, while the agranulocytes are the monocytes and lymphocytes. So they end with cyte. Now, I want you to be able to differentiate between the white blood cells based on the number or a physical appearance. In lecture, we also had to know their characteristics and what they produced and things like that. But for the differential white cell count, that was 6.2 Basophils, you had to be able to identify them under the microscope, so what they look like, their physical appearance, and then also based on what the counts were they normal or not? So you should know the percentages. So as a quick review, the neutrophils are the most numerous. They make up 60 to 70% of your white blood cells. They have neutral staining granules and a multi-lobed nucleus. So like all these lobes, and it's hard to see the granules because they're neutral staining. The eosinophils make up about 2 to 4% of your white cells. They have reddish staining granules and typically a bilobed nucleus. You know, the nucleus has 2 lobes. Basophils are the least numerous of the white cells and make up less than 1% of them. They have dark staining granules, and they have an S-shaped nucleus that's often occluded. It's hard to see sometimes through all the different granules you see there. Monocytes are the largest of the white cell. Notice physically it looks bigger. They make about 3 to 8% typically of the white cells, and they tend to have a large, pale, bean-shaped nucleus and no granules in the cytoplasm. And then the lymphocytes, about 20 to 25%. They're the smallest. So again, I think these three, all of the ones that end in "phil" are about the size of a nickel. The quarter size is the monocyte, and the dime size is the lymphocyte. So lymphocytes are the smallest ones. They tend to have a darker staining nucleus that takes up most of the cytoplasm typically. So an old mnemonic that kind of helped you go from the most to least numerous was never let monkeys eat bananas, which is the first letter of each one of those. So going from most to least numerous, never let monkeys eat bananas, neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So basically what we had to do for the differential white cell count is make a slide, stain it, and then count as many cells as we could and identify each one of those. In a theoretical sense, if you could identify 100 cells, then each one of the cells you found would represent 1%. So you just make a slide and let it dry after staining and go and count cells. It's sometimes difficult because the blood smear that you have to make is difficult to do. But that's, all we have to know for the differential white cell count. So the white blood cell is probably the easiest lab that we have to do in this section. Not very much detail, and a lot of the detail that you do have to know, you have to know for lecture anyway. So you're not learning something new, you're just kind of just remembering it for laboratory as well as lecture.